Hello, buen día, good morning everyone. My name is Javier Gutierrez and I would like to introduce you to this PlasticHeal final conference. It's actually titled Plastic Inside Us, Unveiling the Hidden Impact of Micro and Nanoplastics. We are so lucky today because actually this is the final conference of a European 4-year research project, and that means that we will have plenty of results. In addition, we will have 6 renewed members of the scientific community that will give 5 30-minute talks each that will be followed by a 10-minute question spot for you to ask any concern, any doubt, any idea that you want to comment on them. So one of the major issues of a scientist is actually identifying a problem in the environment and developing a project, actually, that allows its resolution. On PlasticHeal, we have focused this idea into micro and nanoplastics. I would like to introduce you to Alba Bonilla. She's actually an expert of mutagenesis, and that's why she's co-leading the mutagenesis group at UAB, Universidad Autónoma de Barcelona. Her research expertise regards on environmental toxicology and the development of new, more advanced models. Regarding micro nanoplastics, she has studied the chronic effects, their impacts on respiratory tract, their impacts on intestinal tract, their ex vivo exposition, and the development of new nanomaterials. Almost nothing. As if her merits were not enough, she's also the leader of the PlasticHeal project and the leader of the Work Package 6 project that has to do with long-term exposition and carcinogenesis. In case that you have not noticed, she's also my boss. So, well, Alba, I think now the floor is yours, whenever you're ready.
Yeah, thank you, Javi. Okay, well, thank you, Javi, and thank you all for being here, because this means that you care and you are interested in the work and the research that we've been developing in the last 4 years in the context of the European project PlasticHeal, which actually is one of the first large-scale European projects that has been funded specifically to analyze or understand if micro and nanoplastic exposure is really a problem for our health or not. So since the discovery of plastics in the early 20th century, these materials have revolutionized our everyday life and their versatility and durability and cost effectiveness have made them indispensable in many applications and many sectors, such as the one that you have here on the slide. So they are part of some of the renewable energy solutions, like solar panels. They help insulating better our buildings, for instance. They have helped extending the shelf life of food through food packaging, reducing plastic waste, and they are part of a lot of common medical devices and equipment that actually helps saving lives. Having said this, there's a jump from these benefits to this situation where we are now. Their widespread use, actually, I should say their widespread misuse and improper expansion have raised significant concern and plastic pollution. And you have here some numbers that illustrate this. Every year, worldwide, there's a production of more than 300 million tons of plastic, and 40% of these plastics is single-use plastics. If the current trends in using are also in the future, it is estimated that we will double this production within the next 20 years. Since very few of this percentage of these plastics is actually recycled, finally, after producing all this, a lot of million tons end up in the environment each year, causing serious concerns and pollution. But this is only the visible part of the problem that we are facing, because there is an invisible part, which are the micro and nanoplastics, because each one of the plastic items of these millions of tons of plastics that are surrounding us, and I take in here an example of a water bottle, during the whole life cycle, and this means from the pellet production to the fabrication of this bottle to the transport when we use this bottle, when we dispose, recycle, or finally when it becomes a waste, there are several mechanical and biotic and abiotic processes that fragment these microplastics, generating the so-called microplastics and nanoplastics. So, plastics, micro and nanoplastics are everywhere. It's in the food that we eat and the water that we drink, and it's in the air that we breathe. So humans are exposed to this micro and nanoplastics through ingestion and through inhalation. And if we consider those plastics that are smaller, the ones that are below the 1 micrometer, those have the potential to be able to translocate through the gastrointestinal and the respiratory tract, go to the blood, and distribute to the different organs. And if we think in this scenario as a daily basis exposure during all the life, we end up with some evidence that plastics is accumulating in the body. And there's growing evidence that we can find these micro nanoplastics in different organs of the human bodies of the people that have been analyzed. So in this scenario of exposure, we need to understand whether these micro nanoplastics are a risk to our health or not. And this is important because micro nanoplastics are part of an environmental factors that we know that can impact our health. They can cause severe diseases, including cancer, in the example that you have here with air pollution, and it can be quantified. No? And it's thought that the 20% of deaths per year can be actually attributed to these environmental factors. We need to know and we need to understand whether these micro nanoplastics are part of these environmental factors causing these diseases and causing these health problems. But it is not only important, actually, to understand if there is a problem. Scientific knowledge is also important to drive and move forward and to find solutions. For instance, scientific evidence can help enable policies and regulatory initiatives that are devoted to protect the exposed individuals by the hand of policymakers and authorities. It can also drive innovation in alternatives that are safer from our health, where industry is involved. It can also help changing behaviors of all consumers if they are aware of what can be the risk. And they can also be used to protect potentially highly exposed populations, or even to control the managing of patients that we know that can be their disease can be aggravated by plastic exposure. So I hope that those that you are here can identify yourself in one of these potential categories of the different actors that are actually part of the solution we are seeking for. In the end, this scientific knowledge seeks to find what are the safe limits for the population, so we can really use these values to protect the population. This benefits policy progress, this benefits, in the end, a better environment and healthier lives. So it does matter. It does matter understanding the risk, to identify it and to find solutions. But it is not an easy task. To be able to categorize or calculate the risk, we need two components. We need to understand very well how many micro nanoplastics do we have actually in the body, and we need to understand if these particular or specific numbers are hazardous to our health. These two components are combined, and then we can understand if there is a risk or not. But it is not sufficient with some 2, 3, 10, 20 studies that really deal with the exposure or with the effects. There's a long path of growing evidence that we need to accumulate until we get to the point where we can use this to actually calculate the risk. What I mean by that is that this is really, we need years of investigation, because what we need to know here is very specific. We need large amount of evidence. We need to have this evidence in different experimental systems, from cells in the lab to mammalian species that we expose in the lab to actually real exposed humans. We not need to know specific quantities, how much plastic, what types of plastics do we have inside, and where in the body. So these are questions that we don't easily solve. And regarding the effects, is the same thing. We need to have a large amount of different evidence of about many different effects that matter for the regulatory perspective and from the health perspective, such as some examples that you have here. We need this evidence generated in the lab, in the cells, in rodents, mammalian species, in humans, and we need to know very well what is the dose response of this, the behavior of this, what is the dose that is causing the effects that we are observing. So for any environmental factor, following this path until we accumulate sufficient evidence is really tricky and difficult, but for micro nanoplastics research, we have even a bigger challenge, because there are important technological and methodological challenges associated with this type of research, because we are dealing with particles. For instance, in the exposure side, we lack reliable, or we're starting to have reliable and validated methodologies that allow us to measure micro nanoplastics in the human body, especially those that are below 1 micron, that I already told you that are the most relevant in terms of potential health issues. When it comes to the effects, we also have important technology and methodology challenges. It's not easy to have experimental models in the laboratory that are adapted to the exposure to micro nanoplastics. It's difficult to expose them in a way that are significant for real or on how humans are exposed. And we don't have a lot of different micro nanoplastics that we can buy and use in the lab to expose the cells and seek for the effects. In addition of these methodological challenges affecting the exposure and the effects, there's another question that also needs to be taken into account. And is that usually we say micro nanoplastics as a single thing, but in fact, they are many different things. If we see, for instance, this picture where we have different potential plastic items, they are from different polymers. They come, the micro nanoplastic generated out of them may be in different shapes and of different sizes. And moreover, they can be doped with many different additives. So micro nanoplastics is not one thing. There are many many different things, and this needs to be taken into account in the investigation. So we need growing evidence in each one of the particles, shapes, and sizes that potentially can be generated. So it was really a challenge at the beginning of the project, but we count on a big team of talented people that accepted the challenge and tried and started to work on the topic. This team is highly multidisciplinary. It encompasses 7 different countries in Europe and 11 institutions around Europe working on this topic. So, as a summary, and before we start getting detailed information for the next speakers, I have summarized here what was the scenario before PlasticHeal in this path of having enough evidence. We started PlasticHeal at the very beginning. As I told you, PlasticHeal was one of the first large-scale funded projects with this objective. We had limited types of micro nanoplastics to be in a tube in the lab to expose our systems and see what happens in terms of the effects. And we have managed to obtain a catalog of real-life micro nanoplastics that now can be used to screen for the different effects. We didn't know what was the behavior of micro nanoplastics inside the cells or inside the body, and once they face the body barriers of the gastrointestinal and the respiratory tract, and we have adapted a plethora of different experimental models to be able to assess this. And thanks to this, we now know better what are the kinetics, what happens with the micro nanoplastics when it comes in contact with the cell, how it enters in the cell, how it distributes in the cell, and the same with more complex systems like in vivo systems. We have identified some red flags for potential effects for some of the particles that we have analyzed, and you have here some examples. Some have the potential to produce changes in the DNA, problems in the gastrointestinal tract. They can damage the microbiome. They can cause inflammation that can be sustained over time, and etc. And we have also tried to understand what are the mechanisms by which these effects are actually being produced after the exposure. Before PlasticHeal, we had barely no technology able to measure these tiny nanoplastics in the food, in the air, or inside humans, and we have managed to set up all the technology needed for this, and thanks to this, we have generated exposure information on real exposed populations that are heavily exposed to micro nanoplastics in the recycling and the textile companies. And before PlasticHeal, we didn't have tools, in silico tools, to really model those response, to understand what is the link with the dose and the effect that we are observing, and we didn't have any framework to really gather and collect all the information that we are generating and deliver a potential risk assessment information. So this is all the areas where we have advanced, and we can say now that we have moved the field forward. Of course, a lot need to be done to finalize here, understand what are the safe limits for the populations. So over these 4 years, we have been sitting in the same table with different actors involved either in generating the problem or finding the solution. We have been asking them many times what are the questions and the concerns that matter to her, because we as researchers want to do things that are, in the end, return to society and respond to the concerns of the society and the different actors involved. And now we are here not to listen to your concern, but to try to give what are the scientific answers that we have found to the concerns that you have expressed to us in the past. And with that said, I think that what we need to do is to listen to the coming speakers talking about the specific findings, the specific answers that we have found along these 4 years of work. And thanks for your attention. And if you have any question, I'll be happy to answer.
All of these particles are actually inside our human bodies. They can cross our barriers and can affect or pose a health effect for us, a potential health effect for us. I want to ask you just a small question, but I want an energetic response. Do you think that PlasticHeal has actually reached it, or we know more than before PlasticHeal? Just for the record.
Yeah, of course. I truly believe, I absolutely believe that we have moved the field forward and that we have done a lot of great work that can be continued by the researchers that are part of the consortium, but also for others that want to screen for potential effects can use the methodologies and the information that we have generated as the basis.
Amazing. Thank you so much.
Okay, thank you.
Okay. So now we will be moving forward. Yeah, you can give her an applause, please. So now we are moving forward, and we will move to the next speakers. So now the mechanics is like they will make a 30-minute speech, and then we will open 10 minutes question and answers. Okay? So moving forward, once that we have identified the problem, once that we have identified that we need to assess human exposure of micro nanoplastics, we have to say that getting human samples is always a little bit tricky. Detection of micro nanoplastic in human bodies is a little bit complicated. That's why scientists usually tend to use models that approach or get closer to human reality and then test what is happening there and extrapolate the results to humans. I would like to introduce you to Cyril Busy. He's actually the leader of the World Package 4 that tends to fate of micro-nanoplastics within the body, and he is a lecturer at Manchester University. Please give him a warm applause. Thank you so much, Cyril.
Good morning, everyone. So I'm going to summarize very briefly all the results that we have obtained that are relating to the impact on health we have seen across the different models. So I'm not going to focus only on the one I was coordinating, so all the activity related to complex models that are close to human, but going from very simple model up to the most complicated ones, and you will see that I will explain all the different models we have used to obtain those results. So, looking at who cares about that result, first the society, obviously, but all the medical professionals, because if we start to identify that there is a cause for concerns and connection between microplastic and disease, they will be the one that will help us to recover from that disease and alleviate the symptoms. So therefore, that's why we want to make sure that professionals are also on board with us and trying to understand what may or may not be a cause for concerns with plastic. So why should they care about macro nanoplastic? Because just like us in biomedical research labs, there is a lot of plastic around them when they treat their patients. They are using a lot of plastic tools, equipment, and so on, and their protection to prevent direct connection with the patient is also, so the gloves, most of the protection are also plastic-based. So that's why for this particular audience, we believe they are as concerned as the global population and also as concerned as biomedical researchers, because of this plastic surrounding that is all over the place. The first reason, some example of why they should care is because there is some papers that are already showing that within biomedical settings, there is actually potential exposure of those professionals and patients. So that's one typical example of a paper when it's released, and that's what we learned from it by reading it, and that was the setting that they look at in a surgery theater, and they found that there was quite a lot of plastic around. So mostly they were present as fragments, and the two types of plastic that were identified, so in that case they were not nano, so not the smallest one being below 1 micron, but they were macroplastic made of PET and PP, which is two main components of all the tubing that you find in a biomedical setting, and also here and there some tools that are made of PET and PP, which actually you also have at home with all your containers for household product or water bottle and so on, that are all made of these two main polymers that we are using for containing a lot of liquid. The other one is the patient side of it, because obviously you are putting eventually something in the blood of your patient, so that means you are using a part that is metal, but the rest is all made of plastic, and whether or not that plastic is also releasing at that point of injection, and that's the case in there is potential exposure. When you test all the tubings within biomedical settings, you end up identifying that there is quite a large range of amount of microplastic that are released from those brand new tubes. We're not talking about reusing tubes, we are talking about fresh tube that is put in a new patient, and you see that there is a large variation between what you are using. So if you inject glucose or if you inject saline, you have different amounts, but what is really important is that one of the most common plastic that is found is the one used for tubing. So that definitely means that what we use to inject patients release macro nanoplastic, the same as what Asba was spotting out in terms of the typical daily exposure of all society by the food and the drinks as well, on top of whatever is in the air. Now, the second reason why healthcare professionals should care about, because of that societal, environmental exposure that Alba was pointing out, that is happening via inhalation, because indoor, outdoor, there is a clear difference. So indoor, think about your home. Usually you will have carpet somewhere in most houses, so that will release macro nanoplastic over time, especially if you are not regularly vacuuming or opening the windows. So you keep accumulating all the plastic, and everything that you are using that is made of plastic at some point, as soon as it's aging a little bit, especially if you let the sun shining on it, the plastic will start cracking and will start releasing macro nanoplastic. So in an indoor setting, it's even more worrying because that means it's contained. Outdoor, you will have to rely on the wind blowing all the macro nanoplastic, and that's why you see that here there was fragment and fibers, and we have found all type of fragments, sphere, and particles in the air as well, everywhere on Earth where it has been tested. The same for ingestion. You will have all type of macro nanoplastic possible, and there is some products that are higher, with higher amount of macro nanoplastics, some drinks as well, so that's all depending on the container. If you are eating up your food, you tend to actually increase the amount of plastic. And the last one will be via dermal contact, so this one is the most secure way of exposure for any type of compounds upon the one that can go through the skin, because the skin is a very good barrier. But if you have just a wound, obviously plastic may go directly in the wound. So why should we care about that type of exposure? Because we are all exposed, and also because, I'm sorry, a bit too fast, there is no direct evidence so far. So all the papers that Alba was mentioning were referring just to showing that we found macro nanoplastic in human body parts without connecting it to a disease, or there is just correlation between a disease and a plastic presence. But there is no causal link between the presence of plastic and disease at the moment. We haven't been able to link the two together. But what we have found within these 4 years is that there is definitely a red flag that is showing us that eventually that's the sign that a disease may happen if we have all this accumulation of adverse effects happening in the body. So we first started by looking at the literature to have a rough understanding of how much are humans getting exposed. So looking at just ingestion and inhalation routes, you can see the two really important are these two. So that's the amount of particles that you will find per year in terms of ingestion and inhalation. So you see that there is definitely much more particles getting in your body via ingestion. So that makes sense because it's accumulating in the environment, then in the food chain, and therefore because we are eating most of days, we end up, and drinking as well, we end up having much more particles than what is present in the air floating, because that's the smallest, lightest one that you will find in the air, whereas all the biggest particles will definitely go through ingestion. So that's what has been found so far, but that is based on all the biggest particles. And that's what we were wondering, because as Alba has already highlighted, what matters more in terms of effect will be the smallest one. And at that time, there was no technique really highlighting or detecting very efficiently the smallest particles. So that's why the first thing we did actually within the project is trying to develop new type of techniques to actually try to identify these smaller pieces, to know if we can actually expand a little bit the amount, and when we were starting to count what is foundable within human samples, tissue fluids, to try to identify those small particles. So that's one type of technique that has been applied to that type of ideas. So to visualize the macro and nanoplastic, we use a technique that allows visualization and also chemical identification of the plastic. So you combine the two and you are able to confirm that what you see is actually a plastic. And that's here the samples how it looks like when you color code what is not plastic versus what is plastic, and then you eliminate that and you see here just the plastic within that sample. So that's not a real sample, so don't worry, that's not something coming from out of a patient, it's just a typical pilot study where you put the plastic, you put your biological material to try to confirm that your technique is actually working. And that technique that I'm talking about is confocal Raman microscopy, or CRM, that you have already seen in Alba's presentation, but that will come more often in the next one, where they will show you all the application that you can do with it. The other technique that was used is actually fluorescence microscopy, so something we use very commonly in biomedical research, where we put fluorescent tag to shine where we want to look at protein or other type of element within biological compound. In that case, we do exactly the same, but for a foreign body, which is the plastic. So you attach this fluorescent tag on the plastic, and then you see where they are going within the biological model. And that's an example where you have a cell over here, that's the nucleus of the cells, and you have your plastic accumulated here in green, and the white part is actually a doping agent that is present within some of the plastic, just to show that plastic are not alone just polymers, but there is sometimes some additives that are present, and with some techniques you can identify both location of the additives and the plastic. So that's what we believe is going to be very useful in the future, right now already, but even within the future, to help us to develop or advance the field by developing techniques that will support the diagnosis of the patient samples, just to try to demonstrate whether or not there is a link between disease and microplastic present within the body. So, going forward, what we found in human, so that's a case where we look at very specific cohort of patient or exposed populations. So in that case, they are patients of high risk that are presenting chronic kidney disease or CKD. And that's the typical people that are going for dialysis to filter the blood that is not able to filter anymore. And we wanted to know whether or not before and after dialysis there were much more amount of plastic present in the blood. So before, because the kidney is not doing this job, not filtering the blood properly, and after, because it's going through a lot of plastic through dialysis, to know if the plastic use, as I showed you before, was actually adding some things. So that's a typical example of what we were able to identify with the technique I showed before, where you see a lot of different compounds found in the blood samples of a CKD patient, and the highest one was polyester, which is also something pretty common that you find in a lot of clothing, for instance. So that's fairly common to identify those type of plastic in the global population, but also even more so in patients that have difficulty for elimination and filtering. We did the same with another type of cohort of patients, which are the interstitial lung disease. Again, because it's a disease that prevents some function that allows clearance and so on. So therefore, we were thinking that there will be greater accumulation of those. And then anything that I'm not going to show right now, which is connected to the next talk, where we will look at workers that are more frequently exposed to plastic because of their occupation. So that means recycling facility, plastic fabrics, and all this type of occupation where the exposure to plastic is more likely to occur. So what we have done is looking at the impact. So far we have published 49 papers. So that's where it's coming from. Just a joke, because we had a quiz yesterday night and everybody was trying to decipher what was the number, and the 49, so that's why I knew that it was 49. So 49 papers were published so far, demonstrating all range of different impact and all non-impact. You will see that I'm not showing that there is always adverse effect. There is also some good news in the talk, is that not everything is hypertoxic. We need to be vigilant, but so far the evidence are not always showing adverse effect. There is no beneficial effect, just to be clear, but there is no effect. Or at least so far, we haven't seen beneficial effect of exposing people or biological model to plastic. So just a range of models we have used. So from what this scheme is presenting is very simple cells, more complex cells, so that's why you see it's really complex, different color to show you what we do. So we put together all models that are replicating what you have in terms of gut or in terms of lung. So we put all the cells that you would typically find in the organ to try to understand which one are interacting with the plastic and which one are reacting negatively to the plastic. Then there is more advanced models that are recuperating everything in 3 dimensions. So that's what we are using before going to more advanced, fully complex model with way of eliminating plastic. So the rodent model, the Drosophila, which is equivalent to a close to, so that's an in-between between animal exposure and in vitro ones, and the cohort that I already mentioned. So we have this whole range, and each one of them gives us indication about what was going on, and we are all using them in complementarily. So we are not putting things on top of each other. They are all giving us some indication of what's going on, and then we need to see if that translates in what we want to see or want to understand is happening within the patients or the workers. So we also use a range of macro nanoplastics. So we have used some that are the most common one that we found in the daily life of humans. So the one that are used for containing food container, plastic bottles, and tea bags. So that's why you will see a lot of outcome coming from the polyethylene terephthalate, PET, or the polylactic acid. So one is based on fossil fuel sources, and the other one is based on sugarcane sources. So we can also compare these 2 types, whether or not the fact that it's biosourced or a fossil fuel source is actually doing something better or less. And then we have applied different type of techniques, so I'm not going to go into the detail of what we are doing in biomedical researches, but I will try to summarize what we found in terms of the highest level of outcome. So the first is inhalation. So that's the typical microplastic in the air. You have your patient inhaling. So what we look at is in the lung region, within that particular region, which is the air-blood barrier interface. So you have here the air, which is light blue, and then you have the blood here. So that's the red blood cells that are in blue because they are deoxygenized, and when you have the exchange between CO2 and oxygen, you have your blood that is reoxygenated. But that also means that to have that exchange of gas, you have also a very thin layer, and that allows eventually some macro nanoplastics to cross or to interact with the cells that are present over here. So there is few cell types that are really important for everything that also involve in infection. So that's what we look at, are all these cells interact or react to the presence of macro nanoplastic. And when I'm saying macro nanoplastic, most of the time what is actually reaching that region are the smallest ones. So the nanoplastic are the ones that we are expecting to find here, whereas all the other ones are what you find in those big branches here, the bronchi, or even the trachea, where the big particles will eventually accumulate. So what we have found with the simple model is that the PET nanoplastic were perturbing waste elimination and some essential organelles within the nasal cells. Then we have PLA nanoplastic with more advanced model. So again, inducing adverse effect with these cell lines. With the organoids, so when we have this more complex model adjustment of cells in 3 dimensions, we have found that the PET nanoplastic were not causing damage to the mucus, which is the one that is helping you to clear your compounds when they are getting in. So when you are exposed to a fire and all the dust within a room, that's the one that will help you. And we haven't found DNA damage either. Then we go to the more complex animal model. So what we found is that the PET nanoplastic from a transparent plastic bottle, whereas the plastic from an opaque plastic bottle, so the PET one, not the PET opaque PET was actually emitted faster, and therefore that was linked to a better inflammation profile. So that means that whatever remains within the lung is inducing more reaction, which is kind of normal. That's what we see with other type of dust. When we repeat the exposure to nanoplastic, in that case, we haven't found any long-term inflammation. So the same, there is acute reaction, but then it's really quickly going down. Then moving to what we found after ingestion. So again, you ingest, you have different plastic, and then we look at all the different parts of the gut. So with simple model, what was really interesting is that because it's going through the gut, you have this interaction between the food that is digested and then the macroplastic, and you have attachment of the food compounds that are eventually attaching onto the macroplastic. And we found that the plastic that were pre-digested were actually accumulating some compounds on top of the plastic, and that was helping for the plastic to go inside the cells. Whereas plastic that do not contain this digestion product were actually not going that often inside the cells. And when it's going in, it doesn't cause adverse effect. The next one is actually with nanoplastic coming from tea bag, again, not finding any adverse effect. When we move to a more complex model, so that what you see here is the larvae of the fruit fly, so the Drosophila. And when we expose them to PET, transparent, PET, opaque, or the PLA, nanoplastic, so these larvae are just feeding on the macroplastics, so they are exposed repeatedly to quite a lot of plastic. And in that case, we found inflammation, free radicals, and DNA damage. So starting to raise red flag in terms of what is happening for a chronic exposure to macro nanoplastic. Yep. Okay, it's a bit slow. Wondering if the batteries are not going off. So then the next one was actually trying to understand what is happening with your macroflora. So the macroflora that is in the gut, that is helping for digestion, is actually very sensitive to whatever you eat, but also whatever contaminant might be in the food as well. And that's what we have found, is that the PET and the PLA are actually disturbing, in one case in the Drosophila and in another case in the mice. So that was disturbing the typical composition that you have, and toward eventually leading to disease, because when your microbiota is unbalanced, that may lead to inflammation, and if that keeps staying in the body, you will end up with a disease. And then in terms of the mouse model, what we found is that even a single exposure was inducing long-term inflammation, and when you do repeat exposure, actually you start to see much more wide range of effect on the different part of the gut. In some cases, there is recovery, but in other cases, there is more long-lasting effect, and then you see the effect still appearing even 28 days after you have stopped the exposure to the macro nanoplastic. And in particular, there is long-term DNA damage, and that causes concern, because that might link to tumor generation if you keep having DNA damage that are not repaired in the different organs. Then moving beyond the lung and the gut, so you see every time that I'm presenting the blood vessel here, so what we look is at those cells that are component, the ones that are trafficking within the blood vessel, but also the blood vessel cells. So that's what was found here, with a different surface property on top of your plastic. And that really matters in terms of how the cells perceive the plastic and how that may impact positively or negatively the cell's response. So definitely the surface group matters. Then we found no cytotoxicity in other type of immune cells when we were preparing those type of true-to-life macro nanoplastics. But when actually we compare PET and PLA, we found that PET nanoplastic was inducing transformation, so that means genotoxic, but leading to promotion of tumor. And that definitely raised concern as well for potential long-term effect. So my take-home message is that there is evidence that the macro nanoplastic are not completely inert, And in some cases, there is definitely red flag. So in terms of what we need to do is focusing more on the long term, because I've shown you what is more or less the acute and short term. And now the next study will be on the more long term and chronic low dose exposure, mimicking what we are actually in fact exposed to. Then, in terms of professional, now I think we have enough evidence to show that macro nanoplastic should be considered as an environmental stressor. So that's why we need to focus much more on the fact that there is potentially a link between disease and this macro nanoplastic presence and exposure. They are more likely to accumulate in high-risk populations, so that has been shown across the literature, and that's also what we have found in our studies. So that means that we need to be as close as possible to the real-world exposure, which means mixture of plastic with additives, chronic low dose, and the high-risk population, because they will be the ones that are most likely going to be affected by this accumulation. So, at the moment, it's very difficult to say if there is a safe level that we can ingest or breathe in, but so that's why we need to have better method that allow us to really screen, and that will reinforce whether or not there is a safe level, so a dose that we can consider as safe and not causing adverse effect, and all the doses that are starting to show sign of irritation, inflammation, perturbation before we actually reach disease. And that's it for me.
So thank you so much, Cyril, for your nice presentation. Actually, I want to congratulate you because I know that you had many work to summarize all the data because it was a lot. So you mentioned that polymer type actually affects, that there's a risk population that you have tested that may be involved, and actually that there's quite difficult to establish safe levels by the moment. I would like now to open the question and answers for the public. In case that you have any doubt or comment, please raise your hand and I will approach with the microphone. No, it's working.
The thing is, you are not using flow cytometry to measure nanoplastics. Flow cytometry.
Yeah.
You don't use that system?
We do. We do use it quite a lot.
You only said about RAM, but you use a flow cytometry to use.
Yeah. Not for detecting plastic, just to check the healthiness of the cells or phenotyping them.
The other question is you can distinguish with the different kinds of nanoplastics,
Yep.
the origin, the chemical compositions.
Yep.
And the third one is, it seems that the size opens the way to enter the cell using the holes in the membrane, right?
No, it depends.
Inside, except phagocytosis, you have to find, a hole to go inside the cell. But you have to demonstrate that the whole of the cellular membrane.
That's not always the case because we have this protein, lipids, and other component attaching to the plastic, and that's what is helping the docking with receptors. So you have proper endocytosis with the typical ligand receptor type. So actually the particles are getting in because they are getting recognized by whatever has been attached to the plastic.
Excuse me, the last one. Have you tell us that you have, compare, before and after dialysis in chronic disease? Have you measured, the contents of nanoplastics in the liquid after, the liquid on the dialysis? Because you have to find that.
A good question. I believe not.
Because the membranes that you use in dialysis are made also in plastic.
Yeah, I know.
You have to know.
I'm fully aware.
To the plastic filter, and the other one who is linked the contents of nanoplastics that you take from the blood. That's it.
Yeah. Thank you. I think someone will answer your question about what was...
Thank you very much. It's a really good question. And we have proved it. And happily, we didn't find any impact of dialysis membrane on the plastic content in the fluid. So just...
Okay. So that was the answer that I couldn't remember.
Any more questions? Okay. So thank you so much.
I think the batteries...
Yeah.
Okay, so now we have faced several models, and obviously what all toxicologists want to do is to test what's happening actually in humans. So you can imagine that human samples are not always easy to get. So when these samples appear, actually all the toxicologists go to that samples, just fight for them. And it's quite difficult to, or in populations, to find a cohort of people that are actually highly exposed, because we obviously cannot expose them intentionally. Luckily, we have Julia Catalan. She's leader of the work package 2, that is related to human biomonitoring, and she will be providing us some relevant data that she has found because on PlasticHeal project, we have faced, or we have achieved to get samples from plastic working industries. So really relevant data. Thank you.
Okay, good morning. Thanks, Javier, and thanks to all of you to be here today. I would like to start my talk with two important statements that I took from the report of the International Labour Organization. The first statement says that workers may be exposed to hazardous chemicals during all stages of plastic life cycle. That means from the fossil fuel extraction and manufacturing of the plastic through the use of these plastics in different applications to the end of life of the plastics when they are recycled, discarded, or incinerated. And the second important statement is that safety and health impact may be associated to this managing of the plastics. And this can come not only from the additive chemicals, but also from the plastic particles themselves, the micro and nanoplastic, which is the talk of this conference. So some health effects that are related to plastic have been known for many years, but they have been mainly associated to the production phase of the plastic, so mainly related to the chemicals that are used during the production for different things. And the main effects have been related are different types of cancers, cardiovascular problems, lung pathologies, among others. The main problem with these diseases is that at the time they are diagnosed, it's too late. Usually it's several years after the exposure, so you cannot do anything at that moment. But as you can see in this slide, there are also some health problems associated to plastic users. I don't know why in this work they separate the workers from the users, because as we know, many workers are exposed during the use of plastics. For instance, in 3D printing applications, when you use plastic polymers, or workers of greenhouses, they are covered with big sheets of plastics, and due to the weather condition, the sun, the temperature, it can be a release of chemicals, it can be a release of particle fragments, and that can expose workers. Workers of the textile industry, for instance, when they are cutting the textile, the rubber-tired refurb business in the automobile sector, or at the end of life during the recycling or waste management of plastics. And I would like to highlight two of these sectors that are the fabric manufacturing and the recycling companies, because these are the two scenarios that we have focused in our project. So what are the hazardous plastic-related substances? As I mentioned, most of them that have been identified until now are the chemicals that have been used, either the monomers that are used in the production, and the styrene is the classical example of a well-known toxic compound, or additives such as phthalates, bisphenol A, or the polybrominated diphenyl ethers. And these three types of compounds, for instance, are recognized as endocrine disruptors. So they can have really severe consequences for the health. The graph you see there show that among the thousand of chemicals that have been involved, that are involved either monomers, additives, in the plastic industry, several of them have got already a hazard classification, but anyway, they have been classified as hazardous. What we don't know yet is about the micro and nanoplastics that are produced during the use of the plastics. So they could have an effect directly because of the particles themselves, or because of leaching chemical additives, or another possibility is that they could act as vector of other toxic contaminants or pathogens. And for instance, Cyril was, in the previous talk, he was mentioned about a disease, interstitial lung disease, that it was already at the end of last century associated with the textile industry, more specifically with the nylon flock facilities. As Cyril pointed out, we don't know we don't have yet a clear answer whether micro and nanoplastic have really a strong impact in the health. This has been the focus of the 5 European projects that have been worked together in the European cluster CASP, and more specifically of our own project, PlasticHeal. In the occupational settings, the main exposure route is obviously inhalation, and how much the plastic, the micro and nanoplastic can impact also depend on the size. As you can see in this slide, if I'm able to show you, the biggest particle accumulate in the upper airways, but then the smaller particles go deeper in the lungs, in the bronchia, and even at the nanosize range, they can reach the alveolar region. And as Cyril has just described, if they reach the alveolar region, where the interchange of gases with the bloodstreams happen, then they could cross the barrier and be spread in the whole body. So this is what usually happens depending on the size of the particle. With the fine particle, meaning the biggest particle, we have a system called the mucociliary clearance system that is able to eliminate the particle in the upper airways. So most of the particles are eliminated and there is not a problem. And if there is something left, still the macrophages can deal with that. But with the nanoparticles and ultrafine particles, they are escaping this mucociliary system and they reach the alveoli. They overload the capacity of the macrophages and then you start to have problems. There can be release of intermediator, reactive oxygen species production, and the recruitment of the inflammatory cells. And this effect can be even worse depending on the shape of the particles. So for instance, many fibrous nanoparticles have been reported to have a more severe inflammatory effect. So how can we study the effects of micro and nanoplastics in the workplaces? Well, first, of course, we have to define what is the external exposure, what amount of plastics the workers are exposed to. And as mentioned before, the main route of exposure in the occupational settings is inhalation. So we should investigate what is the content of micro and nanoplastics in the air. And then what we need to do is this connection between the external dose and the disease onset. And this can be done using the human biomonitoring approaches. So this consists on taking human samples from workers and analyze different biomarkers, what we call biomarkers of exposure and biomarkers of effects. The biomarkers of exposure can indicate us what is the internal dose, the dose that is absorbed and that is acting at different tissues in the human body. And then the biomarkers of effects are cellular or molecular alterations that appear very early before the disease onsets. Most of them are reversible. So if the exposure disappears, they come back to normal levels. But they are like indicators that if the exposure continues, you may have a potential damage in the future after several years of exposure. So this is what we have done in our project in PlasticHeal. So first, we have analyzed what is the exposure in the air in these two scenarios that I mentioned. The fabric manufacturing industry, plastic fabrics, of course, and recycling companies. And to measure what is in the air, we have used, in one hand, the equipment that is described in this picture, and that allows to measure what is happening in the breathing zone of the workers, but also these stationary sites that can cover a broader area. Then, using several devices, we can measure what is the concentrations of particles per cubic centimeter in the air. But of course, we also need to know what is the nature of these particles. And for that, what we have done is to collect particles in the filters. So we have filtered the air, particles are collected, and then we have analyzed them for the content of microplastics. So that means particles that are above 10 microns. And for that, we have used FTIR imaging techniques, but also for the content of the small particles, the nanoparticles. And as it was described in the previous presentation, for that we have used Raman spectrometry and microscopy techniques. In parallel to this measure, in the same occupational settings, we have been collecting samples from the workers. And this slide summarizes the different types of samples that we have been collecting. As you can see, all these samples are taken with non-invasive or really low-invasive technique. The most invasive sample is the blood collection. And the blood collection was taken with these small vacuum tubes that are used, for instance, when you do your normal healthcare checking for the analysis of blood. There are other samples that maybe can be surprising for you. The buccal cells are taken with a toothbrush. So you just brush the inside part of your teeth, and that's all. And then the exhale breath condensate can be collected like shown in this picture. So you only need to breathe through your mouth. That's the reason we have to close your nose for a while, but that's all. It doesn't require any other special intervention. With all these samples, what we can do is to analyze these biomarkers. So frame in red, you have the biomarkers of exposure. That means the detection of micro and nanoplastic, but also some associated chemicals. And frame in blue, you have the effects biomarkers that we have mainly checked alterations in the DNA, if there is or not DNA damage, the increase of inflammatory cytokines that are also indicative of some inflammation response, or signs of the oxidative stress. So we are still analyzing or finalizing the analysis of all our results, but I would like to give you some preliminary findings of our project. So this slide summarizes the results of the analysis of the air samples. So the concentration, there you have the concentration of the inhalable microplastics, so meaning below 100 micron, in both the fabric manufacturing facility and the plastic recycling company. And you also have what is the size of these microplastics. And what we can say is that in comparison with other studies, the concentration that we have found in the air is higher than in other indoors environments. The videos are showing you what are the classical shape of the particles that we have found in the air. In the upper video, you have a reproduction of the PET, polyethylene terephthalate particles, that were the predominant particles in the air of the fabric manufacturing facilities. In the lower video, you have a reproduction of the polyestyrene particles that were the predominant one in the recycling facilities. In agreement with these results, when we have analyzed the samples of the workers that were in this environment, we have found something like you can see here. For instance, this is the one sample from one worker from the fabric manufacturing company, the sample of the exhaled breath condensate, meaning the exhaled air. And of course, if it was PET in the air, it's normal that there is also a prevalence of PET in the exhaled air. However, from the same worker, when we have analyzed the blood sample, the prevalence of particles is different. Here you have a prevalence of PTFE, that is Teflon, because as Cyril pointed out in the previous presentation, the workers, as every of us, are also exposed in our daily life to plastic through ingestion, depending on the food we eat, depending how we cook this food, also the drinking water, etc. So this is what we can find. I would like to highlight that among the particles that are found in the human samples, there is a good proportion of them that are in the nano-size range, that as pointed out by the previous speakers, they can have more health concern because they can go deeper in the organism. So regarding the effects biomarkers, when we have analyzed a subset of workers from the fabric manufacturing industry, what we have found is an increase of the DNA damage in these workers that correlates also with an increase in the expression of some new biomarkers, because our project is not only using the classical approaches of human biomonitoring, but we are trying to develop also new approaches that can speed up this kind of analysis. But what does it mean? That these workers are going to have a serious problem? No, necessarily. As I mentioned before, these biomarkers are just early indication, let's say alarm signs, telling us that something should be done. They are reversible. If the exposure disappears, the levels will come back to the normal as the controls. But if this exposure is persistent in the time, is when you can have a problem. In fact, from the same workers, we also analyze the prevalence or the rate of chromosomal alteration, that is a more severe effect, and we couldn't find at the moment any sign of that. So what can we do to protect the workers of the plastic industry? Well, the first thing we should do, that is what we have done in our project, is to identify what are the micro and nanoplastics that are present in the occupational environments, and what is the concentrations, the exposure of the workers. Then, of course, we should assess what are the health effects that are connected to this exposure. As Cyril described in the general population about the high-risk group of people that could be more vulnerable, we should also do the same in the occupational setting. So we should identify the group of workers that could be more vulnerable to this exposure, for instance, pregnant women. And based on all this evidence, we should develop occupational exposure limits. That means the limits of exposure that should never be above which workers should never be exposed, because otherwise it could be serious consequence for their health. And of course, we should apply risk control measures to eliminate this if possible, or at least mitigate all these problems. And in order to do that, we could follow what is called the hierarchy of control measures, the 5 categories of control measures that are coming from the most effective one, that consists, of course, on the elimination or substitution of the hazardous substance, to the less effective, that is the use by each worker of a personal protective equipment. But this should be done as the last result. And considering substitution, in the last year, there has been a lot of concern at the OECD level, but also at the European level, about the possibility to substitute chemicals or plastics components that could be hazardous by other possibilities that are safer. So this is what is called safe and sustainable by design approaches. That means trying to design already at the first step plastic that could be safer. One example is bisphenol A. As you know, it was banned some years ago, and it has been substituted by other chemicals. However, it's still unclear, as far as I know, whether these substitutes are safer or not than bisphenol A. And something similar happened with the biodegradable plastic and bio-based plastics. So Cyril was mentioned the work that we have done in PlasticHeal, for instance, with the bio-based plastic with PLA, polylactic acid. And as you saw, it's also unclear whether they are safer than the fossil fuel plastics. And that's all. If you have any question, I will be happy to answer.
Thank you, Julia, for this enlightening presentation. Let me please congratulate you again, your team, and thank the companies that have allowed us to get these samples. I got just a short question before opening the question and answer. In your opinion, do you think that it is possible to select a cohort of population that is actually not exposed to micro and nanoplastics?
I'm afraid no, because as far as I know, even I think that even in the Everest, they have found some plastic particles. But there are tools, of course, to do this analysis. I mean, there are other environmental contaminants that are also spread all over. So there are ways to deal with this problem and try to study it. It's true that with the plastics, we have an additional problem, that when we collect the sample, we use plastic devices, plastic containers, so we can introduce a source of contamination in our samples. And this is something that we are also, so let's say it's more tricky in that sense than to study other type of environmental contaminants. Yeah.
Thank you so much. So now we are opening the questions to the audience.
Perfect, very clear. Oh.
So thank you very much for a very nice presentation. I was wondering, like the concentrations that you report in is particle per something. And earlier we saw the presentation where it was concentrations, microgram or nanogram per something. So how do we combine those two when we try to understand the risks? And my other question is related to, like, I really love the sampling that you did, but you also said something about that we're exposed to multiple sources. So how do we use these individual measurements to derive at the aggregated exposure that we're exposed to as individuals and groups of individuals?
Okay. Yeah. Regarding the first question, yeah, this is a problem also that is in the nanomaterials field. that the exposure in the environment usually is expressed like particles per cubic centimeter, for instance. And then in our studies, we use mass-based units. But I think that there start to be some works where they also report particles per ml, for instance. And of course, the problem is that the smaller particles, you may have higher amount of particles, but with less mass. So in the environmental settings, when there is a mixture of everything, probably number of particles is more appropriate. But we still have to see how then we can combine the units between exposure and effects. And regarding the second question, I would like to highlight that we work at the group level. So for instance, for the effects biomarkers, the individual level is not relevant. We are not seeing usually a huge effect. So what we observe is an increase in the average size. But in each group, controls and exposed, of course, there is variation. So this is something we have to consider. And it's true that in this case, we have an additional problem is that this extra exposure. So what we did in parallel, trying to at least control some of the variables, was an extensive questionnaire to each worker about what are your dietary habits, your lifestyle habits, trying to identify if we see something like higher exposure, for instance, or higher effect, sorry, whether it's really coming from something that is not related to the occupational activity. But at the moment, this is the best we can do.
Any further question?
Okay, thank you.
Thank you. Okay, so now there's a coffee break that will avoid us dying for starvation. Just kindly reminder, we have a poster session and all the presenters of these scientific reports are actually here. So maybe you can take the chance to take a look at these posters and ask any of the presenters or the people associated to that. Okay, so I think that's it. See you at 11:45. And please make sure that you don't breathe or eat many micro nanoplastics during this period of time, please. So welcome back. Have you succeeded in not breathing or eating micro nanoplastics? I guess not. So we are going to start this second part of the session, but first allow me to correct a mistake that I made in the previous exponent. It's not 30 minutes talk and 10 minutes of question, it's actually 20-minute talks and 10 minutes of question. Okay, just to clarify it. My apologies. Well, going back to the focus, on the first half of the session, we have make the point in scientific results that are actually tended to be communicated to scientific environments, meaning healthcare professionals, meaning biomonitoring inside scientific community. However, this second half will be driven into other sectors of the society, because in PlasticHeal project, we have put many effort on developing or reaching that audience also. So one of the things that are actually harder for this issue is keeping actually the scientists outside of their comfort zone. That means that we are usually able to speak and defend our results really well in a scientific environment. But when you move us and try to communicate to other audiences that has nothing to do with scientific community, it's not that easy. That's why in PlasticHeal, we have the fortune to have Virginia Mata. She's actually the leader of Work Package 8, sorry, that has to do with communication and dissemination, okay? She's actually the one that keeps all of us outside of this comfort zone, and actually we really appreciate that, because at the end we can succeed on enjoying sessions like this one. Virginia is also a member of the Parc de Recerca UAB, and well, she's going to give us a beautiful speech about how to reach many, many different audiences within a scientific project. So please give her a warm welcome. And the floor is yours.
Thank you. I need to use my glasses. Because yes, I will read. Silence can be powerful. Yet, it could be hard to interpret as well. But yet, I can probably easily measure the impact of that silence before by simply looking at your faces or checking out whether people are still online following us, or just by asking you, have you felt uncomfortable, maybe interested, curious, bored? Now imagine having to measure something that is also invisible. That's the challenge when you have micro nanoplastics, which are, of course, difficult to measure, as you have learned from the previous intervention. It takes years of research, lots of data to be analyzed, and highly advanced technologies to do so. And that means time. But then, once you have that, then you have another challenge, which is how do you get all of that information to make sense to the other part of the society, to the whole society? That's where communication gets into place. This is my role. And while I am not a scientist, my role in PlasticHeal has been to make sure that the scientific findings got out of the lab and get to the society. This is hard. And I am here to explain the challenges that we have faced, but also let me walk you through 3 examples of how we had tackled that challenge as well. So the first one is, of course, it is tough, it is hard. I've made them come out of their comfort zone, but I'm out of my comfort zone here as well, because I'm not used to being the one explaining science. I am making them explain the science that they do. So one of the biggest challenges has been working with scientists that are really oriented, detail-oriented, but not at all or really hesitant to make strong statements. And I get it. Science is everything, but everything about precision, because they have this fear of they want to avoid oversimplification. This is when I come. But they are always like this. So for me, it's hard because, and for all of people working in communication, it's hard. Because the reality is that when we communicate with society, we want clear answers. Because at the end, you want to know, are my clothes safe? Can I drink from that plastic bottle of water? Am I going to die if I drink too much water from that plastic bottle? We want answers. And of course, we want also to create confusion. So we need to find a balance between the two objectives. The simple answer for that question, are we safe with micro nanoplastics? Are they harmful? Is that we don't know yet. We still don't know. We still don't have enough evidence to know whether they are harmful. But do we have enough evidence to know if they are safe? No, we still don't know. But not knowing is not the same as saying that we don't have a problem. We do know that they are an environmental threat. And building from that, we do have evidence that we have hints that they might be a danger for us as well. So, how do we communicate that uncertainty to the rest of society? Because it's not only scientists that are interested in that. As Alba also mentioned, we are addressing to lots of different audiences. And from the communication point of view, what we did is that we tried to prepare a lot of different resources and activities that were addressed to specific audiences, trying to reach the things that could be of interest for them. We prepared training sessions for specialized audiences. We prepared activities, reach-out activities for the scientists itself, activities for schools, activities for regulators, for policymakers, and for the general audience as well. So let me walk you through 3 examples of those activities to see if they can inspire you as well to reach the audiences that you deal with in your daily life. So, first one, let me change the paper, is our collaboration with the Pelagos Research Institute in Greece. They have been studying marine mammals for decades, and they track whales, dolphins, and other species to understand how human activity affects them. Now, when PlasticHeal was being designed, we had this member of the team, Professor Kostas Kostarelos, which by then was working at the University of Manchester, and he had collaborated with them during, decades as well, and he thought that this could be a really good way to motivate the researchers, especially the young researchers, from the project. So they prepared this reach-out activity, and we collaborated. They joined the expedition in 2022 and 2023. Now, the first year, we also tried to combine their scientific work with the PlasticHeal scientific work, and we did grab some samples. But planning the participation and the collaboration in an expedition is not the same as planning the collection of samples for another scientific objectives. So we leave it as a reach-out activity. And what did we find? Well, they find hundreds of marine mammals, including some that the Pelagos had been tracking during the years. And they also find this dolphin with thumbs that got into the news. I don't know if you've heard, but it was really freaking amazing. Basically, our objective was to give a lasting impression for the researchers themselves. And this was the road. It might sound like a dream. I mean, a week in Greece, sailing in the Mediterranean, chasing dolphins in the summer. But some of the participants of that expedition are here now, today, and they can tell you that it was not really a holiday season. They need to work. They need to do the cruise work all during one week, every day, and some of the nights as well. So it was interesting for them to engage with a different perspective of their work performed in the lab, but trying to connect what they were doing in PlasticHeal with another environment and with another fields of work as well. And let me quote Costas when he says, "The biggest achievement is always seeing the faces of the researchers after the experience and the impact that the expedition and the interaction with the animals has had on them." I know some of the, well, I think I know all of the guys and girls who participated there, and they were happy. So if you don't believe me, you can ask them. You can ask, can you raise your hands, please? The ones that participated there. Okay, so there were 12 of them, and I believe that they enjoyed it. I hope that they had, because otherwise this is nothing. But the point is that that's what outreach is about, bringing science beyond the lab, even for the scientists themselves, and bring it to the real world when it finally is where it truly matters. And then we have this other example, which was a collaboration that we performed with the German, French-German network Arte. They have produced this informative documentary about the impact of micro-nanoplastics in the human health, and asked us, PlasticHeal researchers, as part of the cluster in which PlasticHeal is part of, the CASP, to be part of it. And out of 10 researchers interviewed for this documentary, 3 of them are part of PlasticHeal. And today we have great news, because this is a documentary that was produced during last year and will be premiered in April this year. It's coming up next month. And we have the opportunity to offer you the first introductory video of the documentary. It's in French with subtitles in English. I hope that you are able to follow it, but it's only 2 minutes, and I'm really proud and happy to share it with you. So let's see if it can run easily.
Oh, geez, that's great. Is the lighting halfway decent?
Yes, indeed. They've got the flag up now, and you can see the stars and stripes on the lunar surface.
Beautiful, just beautiful.
Quelle empreinte avons-nous laissée sur la Lune? Des traces de pas, oui, mais pas seulement. Nous avons aussi laissé du plastique. Ce drapeau est en nylon, un tissu synthétique, un tissu plastique. Ainsi, cette bannière ne raconte pas seulement la conquête de l'espace. Elle raconte aussi le triomphe d'une matière qui a détrôné toutes les autres.
Crystal clear plastic that lets you see everything you wrap.
Saran wrap.
You'll love it.
Depuis les années 50, le plastique révolutionne notre quotidien. Pour ranger, cuisiner, décorer, jouer, nous l'utilisons à chaque instant au point de ne plus le remarquer. Léger, modulable à l'infini, peu coûteux, il est devenu indispensable à notre société. Le plastique a conquis tous les espaces pour finalement les envahir, les submerger. Cette matière pollue les mers, les fleuves, les sols, les animaux. Et nous? Et notre corps? Pourrait-il lui aussi être envahi? Au centre de cette interrogation, les micro- et nanoplastiques. Les scientifiques veulent découvrir si ces particules invisibles peuvent coloniser nos organes.
Chercher des micros et des nanoplastiques dans les tissus du corps humain est un véritable défi pour les scientifiques.
C'est maintenant que la communauté scientifique intervient et lance l'alerte. Ce contaminant est-il dangereux pour la santé?
Si on commence à être exposé quand on est tout gamin, qu'est-ce qui se passe quand on aura atteint la cinquantaine?
Avec ces pionniers de la science, nous plongeons au cœur d'un nouveau champ de recherche qui pourrait bien bouleverser notre rapport au plastique. Cette matière, que nous pensions si fantastique, pourrait-elle être toxique pour notre santé?
Okay. So, it looks promising, at least for me. I'm curious about it. I hope you are as well. Why is this kind of collaboration important in terms of communications? Because, of course, it allows us to reach a wider group of people. And because audiovisuals have the power to get to the emotional part of the people, which, of course, is an additional resource when we're talking about science, because we all know that scientific papers are really well and important, but maybe they are not, they don't have that power to get to the emotion of the people. So this is another way to bring the science beyond the lab, because the more people know and are aware of that problem or are informed of that issue, the more people and the better that they can act, of course. And then we have this one that I love. I really like it. How do you explain all of this to 10-year-old? The thing that we did to answer that question is to prepare a digital resource addressed to 6th graders, and we tested it here in Barcelona with the idea of explaining probably concepts that are difficult to understand, but in a way that combined the engagement strategies and trying to do it in a fun way, but also in a scientific way. So we tested it in Barcelona with a group of 6th graders, 17 students, with the participation of 3 teachers, and also 3 researchers from the PlasticHeal project. We made them get together to discuss the content of this educational resource, and we work together with the teachers and the researchers to prepare the content of that, to prepare the story. The kids get to explore 3 major sources of micro and nanoplastics and then try to understand what happens when they enter our body. So it's already available in Catalan, but it will be soon available in English and in Spanish, and it will be openly free of use at the PlasticHeal website. And it also includes an educational proposal that could be used in the classroom by the teachers. And the idea is that it could be adapted and used all around Europe. It was applied in 4 sessions, in two of which the kids had the opportunity to directly work with the researchers and interact with them and ask them questions about anything that come into their minds. For instance, these are some of the images. And I'm really proud to tell you, I hope that they are still online, but a minute before we went into the break, they showed me that they were connected following us, and I'm really, really happy because they were really interested and they really wanted to know how a scientific conference is going to be, and I'm really happy to let you know that they were really engaged with this work, and especially they were really interested in the possibility of having a real scientist being able to explain them or to answering all of their questions. Some of the questions are here. I'll leave you to it. They prepare a list of several questions to be addressed to the researchers. And we were waiting for these crazy days of the conference to pass by so that they had time to answer their questions as well. But I wanted to point out with this work that they were already discussing in the classroom the Sustainable Development Goals of Europe. And they were debating how governments respond to the environmental challenges. So this tool helped them connect the scientific evidence that we have so far with that specific role that the government had. So they practice the strategies that the governments can apply and how they can connect the new knowledge that is being generated by the scientific institutions to make them available to the rest of the society. And this was another strategy that we used for that purpose. I don't want to end up without mentioning that there are two transversal, really important issues that PlasticHeal has taken into account since the beginning. For instance, we've worked with a real interest in including the gender perspective into the research and in the work that the scientists have performed during these last 4 years. We have incorporated a space for, I don't know how to do it. And I'm afraid I will show you that. Okay. Every report that they do about their work, about their research, includes a section dedicated to comments, how they applied, how they tackled the gender issue, even though if it wasn't really the focus of their work. For most of them, it was the first time that they were addressing this in this form. For most of them, it was not. But this is the beginning of, this is a good starting point to incorporate this kind of perspective in research that might not have been thought since the beginning from that perspective. Then we had this gender commission with the support of experts in gender perspective as well. And we also have done things like highlighting the presence and the work of female scientists in the project, and also ask the whole team, both male and females, how they had changed their daily lives, how the project and the work in the workload from the project has affected their daily lives. So this is like some examples of how we had addressed this. And then we had, of course, the participation of the young researchers. Most of them are here today. They have participated in every yearly meeting of the project, but also in outreach activities like the Pelagos expedition that you have seen. And I don't know, we also organized a poster exhibition in one of the clusters meeting last year, and we're organizing the final one as the project. PlasticHeal is organizing and coordinating the final young scholars meeting, researchers meeting in the scope of the cluster. And so, this is like the final part of my presentation. All of this is to say that science without communication is pretty similar to the silence. It's like speaking in silence. It can be meaningful, but nobody could be listening. And the objective, of course, is not to alarm people. It's for all of us to be alert. Because at the end, our goal isn't to create panic. We want to create awareness. And we want people to be alert, not panic. But at the same time, we cannot just say, okay, let's do nothing, because we don't have enough evidence that these things are dangerous. Science evolves, and if we wait for science to be able to offer us definitive evidences of that, it might be too late. So we really need to act, and to act from all over the world, from all over all our roles in the society. If microplastics are everywhere, so the knowledge about them must be, because it is the only way forward for us to act and to tackle this potential problem for society. Thanks.
So thank you so much, Virginia, for your presentation. I think the communication in general, and I include PlasticHeal project, of course, have made a great effort on rescheduling past beliefs and achieving, shifting the issue that micro-nanoplastics were actually an environmental issue into micro-nanoplastics are actually a human issue, because they are inside our bodies. I think all is all of this work is mainly due to communication. So again, and I think the whole audience, want to thank you again for this achievement. I have actually one of these questions, that is, which is the sector that you think that has been easier to reach, and which is the one that has been harder to reach, according to your expertise?
Okay, scientists are really motivated people, so of course they are interested in this issue. They are also reluctant to sometimes sharing the information about their researchers. This makes it hard for us to be able to communicate, because of course there's the issue of privacy, and when you don't really have the results prepared to go out, for me, it could be like, okay, this is really important, we need to talk about it, people need to know, but of course it could be misunderstood, and I understand. Oversimplification could be an issue. But at the same time, scientists are willing to know more, so they are probably the greatest audience for, for us to communicate. And then, in this particular field, I think that society as a big, it's more complicated. Not because we are not interested, but because it's hard to explain that sentence, be alert but not alarmed. It's hard to come up with something that explains what you're doing and tell you, okay, you can do something about it, but just don't panic. It's difficult to reach that message. That's it.
Thank you so much.
Okay.
So we are now opening a question and answer. Please, again, if you have any comment, doubt, raise your hands.
Or don't.
You explained it so perfectly.
Okay.
I guess. Vale. Thank you. So on PlasticHeal project, we have tried or made a huge effort on reaching many different sectors of the audience. However, there's one that is a key, and they are actually the policymakers and regulators. At the end, they are the ones making the law. So they are the ones that can regulate how safe are we when we are exposed to micro and nanoplastics. That's why in PlasticHeal we have put a lot of effort in reaching that concept, because at the end, what we wanted is help decision makers to get a decision regarding these toxics. So I would like to introduce you to Stephen. He's actually the leader of Work Package 7 that has to do with regulatory and policymakers. And well, he's actually as an associate professor at Denmark Technical University. So please give him a warm welcome.
Thank you. Where's the power? That's it. Oh, the power is all mine. Thank you very much. Good. So I'm here to present the Plastic Risk CAT framework, and also why I would say that micro and nanoplastics should matter to governments and local authorities. So basically what I'll talk a little bit about is why plastic is what we call a vertical problem. I'll introduce the framework that we've tried to develop. And then in the last part of my talk, I'm going to address some actions that can be taken despite of the lack of knowledge that many of the previous speakers have alluded to already. So there's no doubt that micro and nanoplastics is a vertical problem in the sense that it's being addressed on every level of governance these days. Like we have on the, I guess, the highest level, we have negotiations going on related to the development or the agreement about having a United Nations microplastics treaty or plastics treaty. The negotiations have stalled at the moment, but they are ongoing. On an EU level, like we have a whole series of regulations that have already been put in place or are slowly making their way through Parliament and the Council in the EU to address various uses of plastics that might lead to the emission of micro and nanoplastics into the environment, and thereby, of course, also limiting human exposure to these particles. So we were in Brussels last week presenting the outcome of not only the PlasticHeal framework, but also this cluster of EU projects that we've been part of the last 4 years that other speakers also alluded to. And here at this event in Brussels, the European Commission or the DG Environment, they actually presented this slide here. It's just trying to give a short overview of all the regulatory processes that were actually already completed or were ongoing related to addressing nano and microplastics and plastics in general. And I just think that like the first one here is related to the Marine Strategic Framework Directive back in 2008. And there's something here from last year where there's revisions to the Urban Wastewater Treatment Directive. So it's just to let you know that regulators are very well aware of the problems that we have and that we've also alluded to earlier, and they're slowly trying to address them for sure. One of the key challenges that they face and that we also face in our understandings of the risks of micro and nanoplastics is the dramatic increase in the scientific publications on the topic. So on this slide here, you can see the development of research publications within nano and microplastics in the past years. And basically in the last 3 years, every year, there's been about 7,000 papers published on the issue. That compared to back in 2009, there was only 300 papers on the topic. So we have now more than 20,000 papers within the area of public health and toxicology within nano and microplastics, according to a search on Web of Science. So it's just a massive amount of literature. And how do we actually keep track of all that research? And how do we also communicate it to decision makers as scientists? What policymakers told us when we started this project is that they would really like us to answer the fundamental question about what are the risks, because they knew that we're exposed. They were alert to the early indicators or the red flags that were like slightly emerging at the time. But basically nobody has really been able to bridge this exposure hazard combination that Alba alluded to earlier. As part of the cluster that we've been a part of, we've published and been part of more than 100 scientific papers and deliverables. We've published work, as alluded to earlier, all along the kind of the body and the life cycle of nanomaterials. And then like the key challenge that we faced was how do we actually communicate this knowledge? So the idea that we have is that we would always communicate what we know about micro and nanoplastics in the form of a radar plot. And I'm going to spend more time in the presentation explaining the different components of this radar plot. But if there's something that I'd really like you to take home with you from this talk is this radar plot. Like when you close your eyes and you think of the presentation I gave, the picture that has to emerge is this radar plot that we use to communicate what we know about the risks of micro and nanoplastics. We've also developed an AI agent that you can use so that you can kind of generate your own radar plots on any given polymer that you can think of, and you can combine it with any kinds of additives or contaminants and so on that you can dream of. Of course, it's an AI, so I will also come back to that a little bit later on, how to actually make the most efficient use of this AI. So this is when you apply the AI to the data that we have generated in the CUSP project, and part of that also the PlasticHeal project. This is the outcome of applying our framework and the AI. So it will evaluate all the different hazard endpoints that we've asked it to, ranging from carcinicity all the way down to endocrine-related effects. Then it will evaluate the strength of evidence and communicate that in, for instance, weak, moderate, or strong. And then it will provide a short explanation for why it has assigned this strength of evidence to that specific endpoint. Okay, so back to what is it that this radar plot actually illustrates. So when you look at such a radar plot, the first thing that you really have to be alert to is the scope of the plastic risk CAT evaluation. So what we suggest is that underneath our logo here, that's where you put in the name or the scope of the evaluation that you're performing. So this could, for instance, be polystyrene, it could be PET, it could be PET plus certain additives, and so on. Here are a few examples of how the radar plots can vary depending on the scope. So up here we have a given polymer 1, where you can just see that the line in the radar plot is illustrating what we know about this specific polymer. We might be in a situation where we would really like to compare the profile of different polymers in one plot. And that's an example that we have here. Here we have several polymers, and then of course each colorful line represents our state of knowledge when it comes to that specific polymer. We also have a lot of situations where different polymers are used in combination with different additives. It's pretty seldom and pretty much impossible to think of that we have a polymer that's used in our society without additives. And the number of additives range from 5 to more than 50. So this is probably the most likely scenario that you'd have. But here you can illustrate what is known about the polymer itself, and then you can also illustrate what is known about the different additives that we know or have suspicion of being in the product that you're considering. And then finally, here we have the option of providing an illustration of what we know about the polymer in combination with different environmental pollutions or pollutants. There's a lot of work in PlasticHeal where we have studied individual polymers in combination with environmental contaminants, and we saw an increased effect of the nano and microplastics. So this is something that is kind of encapsulated by this illustration down here. One of the key things that we discussed a lot is what do we actually put on the category labels here? Because of course, like whether we want to assess carcinogenicity, reproductive toxicity, immunotoxicity, and etc., that's something that we chose as the project. And basically, we have 4 overall categories of effect. We have chronic effects, we have systemic effects, we have organ-specific toxicity, and then we have mechanisms of toxicity. And these category labels are very much inspired by the information requirements that regulators have when industry is applying for permission to produce or import chemical substances to the European market under the European Chemical Regulation. So in that way, we have tried to make these category labels and also the radar plot as meaningful to regulatory decision makers as possible. We're kind of trying to use a terminology that they understand and that they are very familiar with. If you look a little bit closer into the framework, you can see here that we also have a varying level of strength of evidence. So it ranges from very weak all the way up to very strong. And that's an attempt to try to communicate to decision makers and of course the public, like what is the strength of evidence that we have for the given endpoint when it comes to a specific polymer. This is inspired by work and terminology that has been developed by the European Environment Agency a little bit more than 10 years ago. They published a huge volume called Late Lessons from Early Warnings, where they looked back at historical mistakes or lack of use of the precautionary principle by regulators in spite of strong evidence of harm. And what they found is that one of the key issues here is very often the strength of evidence that we associate with the regulation of a given unknown hazard. So they suggest that we assign very strong level of evidence to when it comes to, for instance, proving causality in a scientific setting. But then it ranges down to very weak. And here they have an example of household fire insurance. There might be a very low likelihood that you'll ever have a household fire, but you still pay your insurance every year. So this kind of terminology is what we have tried to adopt and embed into our framework. When assessing the strength of evidence, it becomes a little bit tricky, but there we use a weight of evidence approach. A weight of evidence approach is a methodology that's well established within regulatory governance of chemicals. And basically what it tries to do is to combine all the different lines of evidence that we have into an overall picture. Rather than looking for one study that proves that a certain agent is hazardous, it actually looks at many different studies, the quality of the different studies, and tries to derive a conclusion from what could be a somewhat muddy picture of science. There's some examples of how to do this in the literature, and the European Chemical Agency has also developed a pretty extensive guide on how to assess the weight of evidence, and that's what we've also adopted in our approach. A key aspect of assigning the weight of evidence is assessing the reliability and relevance of the scientific studies. We can have, and as we have, almost 20,000 studies on nano and microplastics, but it does not necessarily mean that we have 20,000 really good, reliable, and regulatorily relevant scientific studies. So there's a difference between scientifically relevant and regulatorily relevant. And when we talk about regulatorily relevant, we mean studies that fulfill certain basic requirements in how they report on the effects that they have studied and the materials they have used, and that can also subsequently be used for risk assessment purposes. And not all science has that specific aim for good reasons. But basically, what we need as part of this approach is an assessment of the reliability and relevance of the studies on which you want to base your plastic risk CAT evaluation. When it comes to the actual evidence itself, there's some rules of thumb that we also follow. Basically, in vivo and in vitro data should outrank QSAR and computational models. Biomonitoring and epidemiological data has the highest weight of evidence. In vivo data outweighs in vitro data. And the results of NAMs outweigh results of cellular assays and so on. So that's kind of like the basic approach that we've taken. So if we have evidence from the highest tier with the highest level of evidence, then we would give that more weight in our assessment. So when it comes to filling in the plastic risk CAT evaluation, there's 5 steps that you have to follow. First, you have to define the scope of the plastic risk CAT evaluation. Then you have to gather all the relevant data and information that you possibly can. Then you have to evaluate the reliability and relevance of this information. Then you use a weight of evidence approach to assign the strength of evidence for each endpoint. And then finally, you fill in the radar plot. And in that way, you can communicate what you know about the polymer, whatever that's topic or subject for your evaluation. So we use this approach to assess all the information that has been generated in the PlasticHeal project, as well as the other 4 cost projects that we've worked with. And this is what the plastic risk CAT framework or application or the radar plot looks like for polystyrene. So you can see here that there are some endpoints where there's strong evidence of harm, where there's a lot of endpoints where we're in the moderate to strong level when it comes to the evidence that we have. We did the same for real-life PET, which is also something that was heavily studied in the PlasticHeal project. And Here you can see that the strength of evidence is not much lower, but maybe down to between weak and moderate pretty much for all the different endpoints. So this is an example of some of the evidence that we put into the radar plot from the PlasticHeal project. This is a slide that I stole from Alba. And basically, it's a visualization of some of the data that was generated related to polystyrene and real-life PET and the endpoint of cousinicity. And here you can see that there's different biomarkers that has been studied and reported on. And Alba, she evaluated the weight of evidence to be moderate because there was reasonable grounds of concern and sufficient scientific evidence. So this is an example of how we took some of the data that we have generated and fed it into the framework and then derived an overall conclusion. I mentioned earlier that we've developed an AI agent that can do this for us and also that you can apply to do your own evaluations. And basically how it works is that in the back end of this agent, we've explained to the agent how the plastic risk CAT framework works, what we mean with weight of evidence, what we mean with strength of evidence, and so forth, how we wanted to assign these. And then using the attachment function that is available, then we attach all the scientific literature that we want the agent to evaluate. So in our case with PlasticHeal, when we wanted to evaluate polystyrene, we simply added all the scientific articles from the project that had anything to do with polystyrene, for instance, and cousinicity. And then we went on to reproductive toxicity and so forth. And that you can also do for the given polymer or the combination of polymer and additives that you want to study. So of course you can rely on the general knowledge that all of these AIs have, but if you really want to make it specific and have it include information that you know of, then you can attach that information to the agent and it will generate that information for you. What we saw over time with using the agent is that, probably not so surprising, but it becomes better and better or more aligned with the opinions of our experts when we add information to the agent. So this is an initial prompt that I asked it to evaluate polystyrene and cousinicity. And it came out with evaluation that the strength of evidence was weak, and it provided a rationale for this. And you can kind of see and recognize some of the terminology that we used when we gave it guidance on how to assess the strength of evidence. So it says here there's limited epidemiological in vivo data. Existing studies focus on additives like polystyrene monomer, computational, and QSAR suggest low direct cousinicity potential. Then subsequently, in the second prompt, what I asked basically is the same question, but I attached 3 publications that I knew of from the PlasticHeal project. And then all of a sudden, or not all of a sudden, but at least the AI changed its opinion about what the level and the strength of evidence is to moderate. And then it analyzed the different articles and made specific references to the articles and explained to us why it is that it now believes that there's moderate strength of evidence. And you might remember that when I showed this slide previously about what Alba's opinion was about the weight of evidence when it comes to polystyrene, it was actually also moderate. So we were quite pleased with the fact that by adding information that we had generated in the project, we could actually make the AI align very much with our experts' opinion on what the strength of evidence is when it comes to, for instance, polystyrene and cousinicity. We love to have your input on the application of this AI agent. And of course, feel free to use it as much as you like. I just want to come with a word of caution that you have to be careful in the interpretation of the radar plots that it generates or the information that it generates. It's well known that AI agents do make mistakes. So you do have to, as it's also specified here beneath, like if there's important information, of course, please double check every time. There's a number of further developments that we are planning with Plastic Risk Cat and moving on also with the AI agents. First of all, like we want to keep developing examples of how to use Plastic Risk Cat for various polymers, various additives, polymers and combined with environmental contaminants. We have been able, and we are at the moment relying heavily on the work that's been done in PlasticHeal and the other CUSP projects in our evaluations of various polymers. And we really want to expand that to the 20,000 papers that I alluded to on one of my first slides, because that's where the use of AI becomes really, really valuable, is that it really enables us to plow through thousands of papers in a matter of hours, and we can put all of that information straight into the radar plots that we're generating. We're also developing AIs that can help us assign reliability and relevance to the various studies. You can imagine having to evaluate reliability and relevance for 20,000 studies is something that would probably take a few decades for us if we had to do it with manual labor and skilled scientists. But thanks to the use of AI agents, we can again do this in a very, very short time. So we want to continue research in that specific field. We also want to add an exposure component to Plastic Risk Cat, and we haven't quite wrapped our heads around how to actually do this at this point in time. But at the moment, when you think of the category labels that I explained, they are all related to hazard endpoints. But as Elba also talked to, and Julia did, like the other side of the equation, exposure is equally important. If there's no exposure, then there's no risks. If there's no hazards, then there's no problem. But we know that there's a lot of exposure, but how do we actually get that visualized in this Plastic Risk Cat framework? What do we want to use a radar plot? Like what do we put on the category labels? What do we put on the axis? How do we assess weight of evidence when it comes to exposure? These are all key questions that we still haven't gotten the real answer to. And then finally, we acknowledge, of course, it's a research project that we have completed, so there will be a need to further development of the GPTs that can assist users in generating their own Plastic Risk Cat evaluations moving forward. Finally, I want to touch upon actions that can be taken despite the fact that we have lack of knowledge. We have, as part of the CUSP project, prepared 3 policy briefs where we have explained both why the projects are relevant for decision makers, what kind of questions they might have that we will have addressed. And in the final policy brief, we provided a range of policy recommendations. And that's based on like what we believe that the state of science is now and what policymakers should do in order to address the lack of knowledge. So the first one we have here is overall strengthening regulatory frameworks, enhanced standardization and certification processes, advanced research and innovation, provide public health and awareness initiatives, and finally, we have a call for the adoption of various precautionary measures. And I think the precautionary measures is something that's especially relevant for you in this audience, like government and local authorities, because it's clear that we have a lack of knowledge when it comes to what the specific hazards might be and the level of exposure, but we have a lot of knowledge about what we can do in regards to limiting exposure. And I think that's where our focus has really been when developing these precautionary measures. Like we urge for restricting the use of micro and nanoplastics in products where safer alternatives exist. Like why would we use them in products where we know that there's a safer alternative? like, despite that we don't know what the actual risks are, like, why would we even take the risk if there's a safer alternative? We want the establishment of guidelines to limit micro and nanoplastic exposure via food and water, prioritizing vulnerable groups such as children and pregnant women. We want to incentivize industry to adopt best practices to prevent the release of micro and nanoplastics throughout the life cycle, from design to disposal. There's way too many products that are being kind of developed without the release of nano microplastics taken into consideration, and that's something that really has to be addressed in order to limit the overall exposure. And then finally, we want to enhance monitoring and reporting systems that enables us to track micro and nanoplastics throughout the life cycle in critical exposure pathways. And that's all initiatives that can be taken without knowing exactly what the hazards are or what the exposures are. And if designed correctly, they can actually spark a lot of innovation within industry and help us protect the public as well as the environment. So with that, thank you very much for your attention, and I'm of course happy to take any questions you might have. Thank you. Yep, you're welcome.
Well, first congratulate, because I think you have found like a pretty good way of summarizing all these scientific data that is actually quite complicated. So congratulations for your radar plots.
Thank you.
You're welcome. You have also assessed that reaching these policy makers is not always easy. So I wanted to ask you, which is the feedback that you have received from the policy makers of this context of PlasticHeal?
Yeah, so we received very positive feedback. So last week, for instance, the Commission, when commenting on our policy brief, said that they supported all the recommendations that we had developed in the COST project. And I think that was something that I was very, very proud of, hearing that. So that was very nice.
Congratulations.
Yeah, that was very good. Yeah.
Okay.
Yes.
So now we are opening questions and answers. Okay.
Did anybody try the AI agent? Not there yet? No. Maybe later.
So thank you so much.
Yeah, thank you.
So now we are reaching the end. We have one extra speaker. And, well, basically what scientific do usually after 4 years of research is collapsing or summarizing all the data that they have collected and answer new questions. Because we always say that for answering one question, you need to face 10 more. That's something that happens in science, and luckily we have Dr. Hans Buchmeister. He is actually the leader of the work package 3 that has to do with impact on biological barriers, intestinal and respiratory. And he is professor at the University of Wageningen. So he's going to provide us the future perspectives and the conclusions of this project. Thanks. Thanks, Hans.
Thank you for a nice introduction. Yes, I have the task and the pleasure to try to send you home with some take-home messages. What do we need to do in the future to really try to address and increase our knowledge on the safety and potential hazards and risks of micro and nanoplastics? Because we are facing a problem, and the problem is perhaps indicated here. Our planet and the planetary health is currently at risk. As you know, there are quite some boundaries or effects that really threaten the well-being of us as humans, but also the system Earth. Climate change, we all know this, that increasing temperatures is affecting how we live and how we can maintain ourselves as a society. But there are also a category that's called novel entities. And novel entities are those chemicals, plastics, materials, micro and nanoplastics, produced by us humans that is affecting the health of the planet and the health of us humans living on this planet. Chemicals, plastics, and microparticles. These are quite diverse classes of chemicals and particles, so it's not one size, one shape, one polymer type. So we're facing with quite a problem, as has been indicated and illustrated earlier this morning. And how can we now study the safety of these particles or identify and characterize the hazards? For that, we use a great range of different models, and you've seen them passing by in earlier presentations this morning. From animal studies, different species have been used to reveal the mode of action and how they interact with whole complex organisms. Us as humans, but it's not always easy to do studies in humans. There are ethical concerns, and it's also sometimes difficult to really identify effects in these humans. So therefore, we also develop cell-based models. And it's at the interface and the interplay between all these different models that we try to increase our understanding of how particles, micro and nanoplastics, interact with humans. And I think it's key to not only screen for the chemicals, but try to increase our knowledge. To increase our knowledge on how these particles interact with biology. In other words, to study the mode of action of these particles. And to do this in a structured manner, we in toxicology have developed this principle called adverse outcome pathways. And it might sound complicated, but I think it's easy to understand. Think that you are exposed to a material, a particle. It reaches a cell in our body. The cell gets alarmed. It might get infected, challenged, stimulated. But our body is not one single cell. In our organs, we have multiple cells that work together. In a tissue, a lung tissue or an intestinal tissue, you have different cell types. So if one cell gets affected, that's not so cumbersome perhaps. But if multiple cells are affected, then it might affect the organ system. If the intestine doesn't function, the health of an organism might be affected. And that's, in essence, the adverse outcome pathway. Think of the Newton cradle at the bottom of this slide. A bullet, you release it, it hits the next bullet. The signal is transmitted, and in the end, the last one jumps out. Exposure, effect. That is the adverse outcome pathway. If we now understand how all these different nodes interact, we can very detailedly study the mode of action of particles and learn more how it works. We use different models for this. Simple models to study the early interactions, more complex models with different cell types, stem cell-based models to really emulate how tissues are in our body in a laboratory. We aim to create so-called mini organs, but also intact organisms still are important to really study and find out the mode of action of these particles. And I will show you a few examples of the models that we've developed. Simple models, easy to use, to screen a lot of different materials. We engineered cells, so that if they're in contact with materials, they emit a light signal. That's easy, because then you can read out if the cells are in contact with the particles. And you know if they might cause an effect at the cellular level. And you see a graph where we screened quite a few particles with different concentrations, and then can prioritize those particles that we really should dive into, because they are potentially interacting with us. This is a simple model, the early step of mechanistic action. But as I said, our body is more than one cell. We have important barriers. We have heard that we are exposed to particles via the air. These particles might hit the lungs, the lung epithelium. The lung is a complex organ. Different types of structures in different parts of the lungs, and we have created, not in my lab, but in the project that I'm part of, different models for the lung. You see nice colorful pictures. The different colors represent different cell types in these models. So we can study the interaction of particles with the different cells. If you zoom in a detailed way, you see these green cells. These are specific cells called macrophages that swallow particles, remove particles from the lungs. What happens if you have these cells in your models, or if you don't have them? Then we can study the consequence of the exposure to lung tissue, and in the end, try to extrapolate these findings in these mini-organs to intact organisms. Similarly, we can do that for the intestine. The intestine, equally to the liver, to the lung, is composed of different cell types. So we created models, you see that in the top panel, models with different cell types. Cells that are important to take up nutrients, cells that secrete mucus, a protective layer on cells, and regulatory cells. We expose them to particles. And we would love to know, are these particles present? So we have sensitive analytical techniques. You've been hearing about these techniques earlier today. And we can confirm, look at the red colors here, that these particles are in the cells. So there really is an exposure, not only to the outside, but also in the cells. Do they cause harm? Well, they do, but not all. So here you see 3 graphs with 3 different endpoints. Is the quality of the epithelial layer in the intestine affected? That's the first graph. And sometimes it is. If the bar is high, there's no effect. If it's low, there is an effect. Are they causing so-called oxidative stress, an alarm signal to cells? Or is the exposure causing inflammation? Again, you see the height of the bars different, which means that for different particles, you see different effects. So not all particles are toxic. Some of them are more toxic than other cells, than other particles. But it's not only the particles as we produce them. In the environment, they change. They change form, they might change the size, but also the coverage of the particles might be different. The same particle in food might look different than a particle in my gut or a particle in your cells. The so-called corona, a layer of proteins on these particles, affect how our body sees these particles. So we need to understand this. And it's important, look at the graph. Again, dark bars showing high uptake, white bars lower uptake. The difference between these particles is the layer of proteins on the particles. So the environment where the particles have been in is important to understand their uptake and their effects. Not only simple particles, but a complex environment is important to take into consideration. And all is fine with these models. We can understand now how they work, but in the end we are interested in effects in humans. So we go in society to factories, take samples, assess the presence of particles or potentially the effects. So that's called human biomonitoring. And that's, I think, very important to continue doing that, to also bridge between the work in the laboratory and in the field in our society. For that, we need very sensitive techniques, very sensitive analytical techniques, and you've seen these pictures before. The example is confocal Raman, and there are other approaches where we use complex technology to screen for the presence of particles, again indicated in red in these slides, in cells or in lungs of mice. But we can also use these techniques to assess the quality of our food in milk or in other complex biological environments. Beautiful techniques, but very labor intensive. And if you want to screen in a lot of different food commodities or in a lot of tissues, we would love to have techniques that can be used more easily or more rapidly. So then we know the exposure. We went into society to screen for the presence of these particles in humans or perhaps some effects. But there's an end to that. So we would also like to create computer models to predict if we are being exposed, as we are on a daily basis, to micro and nanoplastics, where do they go? Not only perform measurements, but also try to predict them using computer models. This is called pharmacokinetic models. And you see a typical cartoon here of an individual taken into a computer model with the different organs. You might recognize the lung and the brain and the heart connected with the bloodstream. And then you can use these models with, of course, input from some data and physiological information to predict concentrations of materials in different organs. In these graphs that you see some tissues prediction of a given particle over time. So you see, for instance, in the liver, there's an increased concentration of the particles in time. Luckily, in this example, there's a maximum concentration. This is important because now we can predict concentrations in the liver. And if in my laboratory we are doing studies with liver cells, we could now say, well, what are the concentration that we need to put on our cells, on our liver cells, to see if there is an adverse effect. That's an example of how we can use these computer predictions, bless you, on the concentration of particles in the tissue and how to make our studies in the laboratory relevant. That is what we call extrapolation from in vitro data to in vivo data. Having a response in our cells, using these computer models to predict concentrations in different organs, to in the end predict safe concentrations, safe doses of chemicals, of particles in air, in our food, to protect human well-being, to set safe exposure limits for micro and nanoplastics. For us as a consumer, for workers, and also for species living in the environment, the approach is similar for all different organisms. So wrapping up what I think that we need to do in the future as a summary of the stories that you heard earlier this morning. We should stop testing one polymer type. And of course, in the project where we show some research highlights, we have done that. And we've shown that for different types of particles, different polymer types, as we scientists call them, polystyrene particles or PET particles, we see different effects. So it's important to differentiate between the polymer composition and the sizes of different particles. In the environment, particles are different than in a factory where the plastic is being produced. So we need to have true-to-life, realistic particles to be studied in our laboratory. What is the exposure? We need to know the realistic exposure to make sense of our data in the laboratories, but not always. Toxicologists love to use high concentrations because then it's easier to find out the mode of action of materials. So there's always a dilemma between what concentration you want to use, but it's needed to know what the real human exposure is. Do we know the effects? We have seen passing by quite a few endpoints, but it might be that these particles are so unique that they cause endpoints that we are not so much aware of. So I would love to study with my colleagues, are there effects that we do not know yet? Do we need to develop new methods in our laboratory to screen for the potential effects of these particles? And there is a concern, of course, because we are exposed to a lot of different chemicals, a lot of different particles, and we do see an increase of all kinds of diseases that likely not only have a chemical cause, but for sure chemicals and particles do contribute to the effects. I highlighted the human exposure, so obviously we need to know more about that. And what's now more important, the high incidental exposure of workers or a low-grade exposure, but over long term for hopefully the majority of us? We simply do not know. There, epidemiological studies in humans are needed. Animal studies with flies, with mice are important, but also these cell-based studies that I showed you earlier, and the integration of all the information in computer models. Can we observe effects in populations? Do we know what to look for? Are we really needed to look at particles, or can we find so-called biomarkers, which are sometimes easier to detect? And we cannot test everything. So we need to be smart. We need to understand, in my point of view, the mode of action of chemicals or particles. And then we can, if we understand the relation between the properties of particles and the mode of action, we can extrapolate our knowledge and observations from one type of particle, one type of chemical, to other particles. What is at stake? I think planetary health is at stake, our health and our well-being. And I'm going to end this 15 minutes or so to read to you a quote which inspires me as a biologist and a scientist, which I took from the book Our Stolen Future. Warmly recommended to all of you to read it. It has been published or written by Theo Colborn already 30 years ago. And at that point, she was really concerned about endocrine disrupting chemicals. That's why the title of this book, Our Stolen Future. And in this book, there's a quote which I think was important at that time, but it's surprisingly, annoyingly, still very relevant for us today. So in her book, she writes, as we work to create a future where children can be born free of chemical contamination, our scientific knowledge and technological expertise will be crucial. Nothing, however, will be more important to human well-being and the survival than the wisdom to appreciate, however great our knowledge, our ignorance is also vast. In ignorance, we have taken huge risks and inadvertently gambled with survival. Now that we know better, we must have the courage to be cautious, as our stakes are very high. We owe that much and more to our children. And there my microphone goes, and this was the citation of Theo Colborn. And with that, I would like to close my words.
Thank you so much, Hans, for your presentation. You left me without words. It was everything so clear. So now let me ask you one more question. With your expertise, what do you think that we need to drive now the focus of attention? What will be the priority to study?
Is to save and to protect really from these unknown effects, and therefore I think understanding of the mode of action of how particles interact with us humans, with cells, but also with other species in the environment is key for understanding and moving forward.
Thank you. Any question?
Yeah, you told me.
I don't know if you know that the specific point in the intracellular systems that are engaged with nanoplastics, the mitochondrial system, is the endothelial reticulum, or is the whatever. You know, do you know this specificity or a simple problem of volume? I have a volume, I interact with another foreign particles, and this is a kind of mix of everything. Or there is a specific target inside the cells.
Yeah, so I think your question, if I understood it correctly, is there a specific target, a specific molecule perhaps that affects us by the exposure to particles? No, I don't think that's really a specific one type of molecule that's affected.
It's only a problem of volume?
Sorry?
It's only a problem of volume?
No, it's not a problem of volume. In biology, everything is related to surfaces. So chemistry takes place at surfaces of materials. So that's what we also see here. So that's why these particles are so intriguing. The smaller you go, the larger the relative surface area is. And as chemical reactions, biology takes place at surfaces, that's what happens. So different surfaces or chemicals bound at the surfaces or different charges, they intervene with membranes and with cells, could cause oxidative stress and all the subsequent steps, potentially interactions with DNA or inflammatory pathways can be affected. So it's not one specific endpoint, in my view, but quite a few, generalized one. But yeah, the particles might end up at different places in our body, different places in our cells than where chemicals would end up. Other questions? Who dares?
Yep, I dare.
Good.
Yep. I was wondering, like the call that you have for us knowing the mechanism of action, but also what you said about like the polymers and the numerous additives that are being used. Like, how can we bridge that gap? Because you also ended up with a call for action. So can we take action without knowing the mechanism of action for every combination of additives and polymers out there? Or what is it that, what you're thinking about
Well,
that?
perhaps it relates to where you ended, the precautionary principle, that in the absence of having solid information what to do, what action to take, and perhaps I sympathize a lot with your concluding remarks. Well, I'm freely interpreting it perhaps, but let's try to reduce the use of plastic or adequately recycle it so that we can reduce the exposure. Not that I'm overly concerned, but it's clearly something that does not belong in the environment, so let's try to not put it there. Yeah, and the chemical exposure and complex mixtures, combination toxicology, complex mixture toxicology, we've been mentioning that in the field of toxicology for decades, and the reason why we still mention it, because it's so difficult to study. Which combination of particles or chemicals do we need to take? There's not one smart step to take, I think, but I would propose that we start to work and start exploring combinations of particles and chemicals. Yesterday we've heard examples of stories with particles and heavy metal, as arsenic, and the co-exposure causes a stronger effect than the single exposure. Well, I think these kind of studies are needed, not only with heavy metals, but also with chemicals that we know that can interact. We have to explore, and hopefully we'll learn soon how to do that more efficiently. There's time.
I don't know if you know, you show the concentration of the different organs. Do you have any idea why the liver, the brain, the different organs have a different kind of concentration? It's because the possibility of transmembrane circulation is different. It's because it's linked a lot of quantity of phagocytosis in every organ. Do you have an idea what the difference in concentration? Because I saw the liver, of course, the liver is a filter, but it is a high production proteins. But do you have an idea between the different organs?
The expert is sitting behind you, but if I try to answer this, so for chemicals, we know that some chemicals like to be in a fatty compartment or perhaps in a watery compartment. So based on their chemical properties, we can predict if they would prefer to be in a fatty environment or in a lean environment, in a liver or in fatty tissues. For particles, we do not know this knowledge, but we know that there are different pores or openings in the blood endothelials, between endothelial cells, that allow the migration or the migration of particles to different tissues. We also know that some tissues are high in cells that swallow particles, macrophages, like in the liver and the spleen. And with this really fundamental basic knowledge about physiology and the structure of different organs, we can explain, at least in part, why some organs have a higher concentration of particles, which is size related, than other particles or other tissues. So looking at the physiology and the properties of the endothelial wall of the blood capillaries and other really fundamental biological principles, we can partly explain your question.
One thing, another question. But have you really make a research in this kind of cell, hematopoietic cells, who are very active, are producing a lot of proteins, a lot of cells? Do you have the sample of this kind of cell?
You have to move up your microphone.
Yeah. If you have a sample, if you have a research about the hematopoietic cells, who are very active, who have a very high level of production of protein of cells, and I suppose if linked with the metabolic activity, that would be a perfect target to talk with that.
So I'm not sure if I can fully answer your question, but what I know that in the past 4 years we have been screening, and colleagues in the project also have been screening really different cell types, from macrophages from the lung to epithelial cells and other cells, and compare the responses of all these different cell types, looking for instance at the gene expression or protein expression. And that's interesting to really dive into, because then we are able to answer your question. Which cell types are now more responsible for an effect in a tissue, in an organ, than other cells? Some, in our intestine, I know that it's full of macrophages. Macrophages swallow foreign materials. Closer to the epithelial layer, they have different properties, different functional properties than deeper in the intestine. And that's important because they have a specific function at specific locations in the intestine. And that works similar for other organs. So it's, if you really zoom into how, with different cell types, how they interact and deal with the exposure to chemicals or particles, yes, they have different functions, they have different roles, and the role and the location where they are follows their function. So it's obviously that not all cells are alike, and it's interesting to really, in a scientific exercise, dive into the different responses of different cell types. We are closing.
And well, I'm afraid also that this conference has arrived into an end. Thank you so much all for attending. Thank you so much the ones that are at home for taking the time to come here and also for this amazing discussion. Well, I also wanted to thank PlasticHeal team because I've been part of it and I think that all of them are magnificent. So I will take 1 minute just to thank all the brilliant scientists that are within the project and all the brilliant human people that are behind the project. So I think that's it for it. I don't want the image of this conference being me, so we'll leave the floor to all the senior researchers that have been involved in PlasticHeal. If you can please approach to the stage, it will be great. Thank you so much.